So this is going to be a little bit different than what we had planned. If anybody's ever been to one of my presentations before, I've got lots of colourful pictures of microbes and colourful graphs and things like that. So this is going to be a little bit different. But thanks to Chris, we've got some butcher's paper and some colourful pens. So while it won't be as good as the PowerPoint, we can still do some good things. And maybe this is, I don't know, it may be really interactive. So. But anyway, so just a little bit about myself. There's quite a few faces that I haven't seen before, people I haven't met before. So my name's Ash Martin, and I run with my colleague and a few other people a laboratory in Adelaide where we specialise in measuring soil microbes and also interpreting the information to help people make better soil management decisions and improve soil health. And so some of this information that I'll present today comes out of a couple of years ago now, or maybe a few years ago now, sort of looking, and we've been to, perhaps like a lot of people here, a lot of soil health workshops and regenerative farming workshops. And there are a couple of things, great days. Usually they were great days, a lot of good information, but there were usually two things were often missing. One thing was measuring it. How do we measure this thing called soil health? And what is it for a start? Because it tends to be one of those sort of generalised sort of conceptual terms. What is it? How do we measure it? And then putting it into practice. And so we came up with this thing called the Transition to Soil Health program, which is really just a framework to give people some basic knowledge about defining soil health, how you measure it, and all the processes that go into that, how you can measure it, and some of the things that you can do about it. And it wasn't dogmatic. Organic practices could fit into it. Regenerative, sustainable practices could fit into it. Anything could kind of plug into it. So it was really a framework. So some of the info today is from that transition to soil health thing. So just to start off, I spent a bit of time preparing researching this, so I'll start off with this. Are there any Hebrew speakers in the room? Are there any Hebrew speakers? No. This is the word for. I was asked to speak on a topic around the living, breathing soils. So this is actually a Hebrew word, ruach, which means breath. And so there's also, for those of you who are interested in languages, there's, I can get this back. What have we got here? We've got, some people might recognise this. Oh, that's a bit rough. That's Greek, pneuma in Greek. So that means literally from we get pneumatic, so also in breath. So that's breath in Hebrew and Greek. And so when I was asked to talk on living, breathing soils, I was thinking about breath. I was immediately thought of the story of the creation of man in the Bible, where God formed man from the dust of the ground and breathed into him the breath of life. And I was thinking actually quite a fitting thing that was for an organic conference, because according to the Bible story of creation, we're all created from the dust of the ground with the breath of life. And so hopefully as we go through this today, we'll see some of that connection. And I know organic people like a bit of philosophic stuff, so hopefully that's my contribution of philosophy. And so really life in the soil is soil health. So you can't have, somebody explained it to me this way once, you can't have a healthy rock, for example. If something's not alive, it doesn't have health because it's not alive. So just by definition, when we say soil health, that implies that it has life. And so things that have life, generally speaking, have breath. But what do we mean when we say, soil life then implies soil health and vice versa? But what is soil health? It's a term that gets used a lot and probably often in a vague way. Would anybody like to, I'll put some people on the spot maybe, because probably nobody's going to volunteer. Say from the far table over there, can anybody give me their definition of soil health? There's really no right or wrong answers, so don't be worried, but what would your definition of soil health be? Yeah.
For us, what we've found is a key driver towards soil health is organic matter.
Mm-hmm. So key driver towards soil health, organic. So a soil that's got lots of organic matter in it?
It's very complex. I mean, obviously.
It is, yeah.
I'll just put out organic matter.
Yeah.
Put that out.
Yeah, organic matter. What about from this table here? Aeration. So front table here.
Supports healthy plants.
Yeah, supports healthy plants. Yeah.
Biology to act on to convert these
Okay.
bio.
It's got lots of biology in there. Yeah. What about this table here? Good water holding capacity. Yep. Back table over there.
Nutrients, the nutrient spectrum.
Nutrients are
On the soil test.
in balance, or a wide spectrum of nutrients.
What you let short of or excess in.
All right. Well, I suppose from that table, maybe I was sitting at that table, so maybe it's my turn. I'll answer. I'll say what. So I thought I'd spare you. So a definition of soil health, incorporating all those things, and this was I spent a lot of time reading and looking at published works and books written by people who are experts in soil health, and also from our own experience. So my definition of soil health, or a definition of soil health, would be the ability of a soil hope you can read my writing. Soil health, ability of soil to, the first one was keep producing healthy plants. Thank you, Tim. While effectively supporting all the organisms, including all the animals and people. We're important too. That depend on it. Does that make sense? Do you think that captures the spectrum of it? And so, but of course, the challenge in agriculture is we want to maximise productivity, however we define that. Of course, we want to be productive, healthy soils, productive soils. So we want to maximise productivity and be profitable as well. That's also important. Thanks, Kim, pointing that out earlier. Maximise productivity while providing for these important soil functions for agriculture and the environment. I have to do that together, and there's a tension in that. Those things are often in tension. And so there's lots of properties of a healthy soil, and as I went around, most of you are mentioning those properties of a healthy soil, like a deep root zone, high soil organic matter, high moisture storage capacity, etc. So there's probably about a dozen or so. Experts in soil research have probably put together a list of about a dozen or so soil properties that you've pretty much covered them there. So I'm pretty confident that you all know those. Anyway, so we're up to this part here. So each of those soil properties, which you pretty much all mentioned there. So we've got a list of them there. And by the way, this isn't Ash Martin's list of properties of a healthy soil. This is from a combination of published research and so forth that's been on the topic. But each one of those soil processes is the result. Each one of those soil properties, is the result of four main soil processes. So we've got residue digestion, nutrient cycling, soil structure maintenance, and pest and disease regulation. So every one of those properties of a healthy soil that you mentioned, and some of those that you didn't, that are up there, come out of these 4 soil processes. And so what's interesting about these things is that they're processes. They don't just happen, they need something. And can anybody tell me what those processes need to work? Life, exactly. So those processes need life. And where we have life, we have breath. And hopefully I'm doing a good job of drawing those themes together. And so all of those things, those soil processes come out of 3 main soil components. So we've got the physical aspect of the soil, which is the structure. So we need the structure, that's the foundation. So we need a structure, we need a home for that life to live in, for example. We need a structure, and that also refers to, for example, the geological background of the soil. So if it's, from basalt or granite, whatever. And so from that we get nutrients, we get some minerals and nutrients. So out of that structure comes nutrients. And then where we've got some structure, a physical home, a physical space, and we've got some nutrients or some food, then we can start to have some biology, some things that are living in there. So that's the way that those kind of 3 components of soil build on each other, and they also all interact together as well. So in this diagram here, you can see we've got on the left there, you've got our 3 soil components that we just talked about. So we've got the structure or the physical aspect of the soil. We've got the nutrients or the chemical aspect of the soil, and then we've got the biology or the living part of the soil. And that includes plants, by the way. They're also living things, not just all about microbes and things that we can't see. And all of those things interact together, and when they interact, they, as a result of that interaction, they produce those 4 main soil processes. So we've got residue digestion, the breaking down of organic matter. We've got nutrient cycling, so the ability of, for example, nitrogen to be fixed from the atmosphere and made available for plants to be uptaken. The solubilization of phosphorus from minerals in the soil to be made plant available and available to microbes and other biology as well. Soil structure maintenance, the bringing together of the inorganic non-living parts of the soil into some sort of order and structure where those living components can find a home and live and do their job. And then pest and disease regulation as well. So keeping the whole system in balance. So we'll probably never be rid of pests and diseases, but they can be regulated when we have a good, healthy, functioning soil system of those things in balance. So probably one of the things that often get asked most often to talk about is biology in the soil. So what are we talking about when we think about soil biology? Well, here's a diagram of what's called the soil food web. Most people would have heard that. And so we've got plants, we've got all sorts of creatures that are in this, what they call trophic levels, orders of, a bit like a food chain if you like. But since we're focusing on soil and soil health, we'll just sort of zoom in a little bit just to focus on that underground part. And so we've got organic matter that's being eaten by your primary consumers like fungi and bacteria, and then those organisms are in turn being eaten by other organisms who are a little bit more complex, like nematodes and protozoas and springtails and things like that. And those things are being eaten by other organisms, and those organisms are being eaten by other organisms, and so we've got this chain happening. And along this way, we've also got the opportunity for nutrients to be released. Every time something eats something else, there's the opportunity for something to be released, some nutrients to be released, nitrogen being a classic case, because these things are relatively concentrated in nitrogen, and these things tend to be less concentrated in nitrogen. So every time something here something further down this chain eats something up above, there's a potential to release some nitrogen and other things as well. So that's one important way that nutrients can be cycled by soil microbes. So what about today? We're going to mainly focus on soil microbiology. So we're really talking about this area here and maybe a little bit into here. And so what is soil microbiology? Well, really a lot of people feel that it's a very complex topic, but so just to try to reduce it down to bite-sized pieces that we can understand, really it's just organisms that are too small to see with the naked eye, and they're various types. In a list there ranging from things like bacteria, which are called prokaryotes, and by the way, don't feel that you have to remember all of these terms. It's just to let you know that these things are studied. This is what scientists call them. You don't need to remember this in order to be able to be a good manager of soil, but just to let you know that they exist. So things like prokaryotes, bacteria, archaea, that's not the Swedish furniture store, by the way. Then there are eukaryotes, fungi, protists, and all those things. As you go down that list, they're getting more and more complex as we go down that list. But that's not to say that even bacteria we often refer to all the simple bacterium, which was a kind of a leftover from the days of early microscopy, where microscopes were, rather crude as compared to today and the technologies that we have today. And so bacteria are thought to be just like little blobs of protoplasm, just a little like jelly sitting there, little circular patches of jelly. But what we know today is that even the simplest bacteria has a system of organs and communication systems within it that would rival the most complex and biggest human city. If you could think of, for example, New York City with all its transport systems and communication systems and people and different roles being played by different people and buildings, even the simplest bacteria would look more complex than that. So when we say a simple bacteria, we have to keep that in perspective, that it's a relative term. And so where are these organisms? Well, they're everywhere, but they're most prevalent in what's called the rhizosphere, and that's just a fancy scientist term for the root zone. And so there can be up to 100 times more microbes in that zone that's about 2 to 5 millimetres around a living root than outside elsewhere in the bulk soil. So that's really where it's all happening. So sometimes people ask, what's the best way to improve soil microbiology? And I often tell them, the answer's right there. Can anybody guess what I would answer? Roots. Have a living plant root. You're going to have 100 times more microbes around there than in the bulk soil. So there's a practical management tip. If you're looking to improve soil microbiology, a good place to start is have some plant roots there. And of course, there's different types of plant roots that we can use to achieve those purposes. And so how does this all work? Well, really all this is based on different types of microbes as well. For example, we've got bacteria, we've got different types of fungi. This is a diagram of what's called mycorrhizal fungi, which I think a show of hands earlier indicated that most people were aware of that was. And they can extend out beyond the root zone if there's a zone of nutrient depletion around roots after they suck up nutrients after a while. These mycorrhizas can grow out beyond that nutrient depletion zone and get nutrients and bring them in and tap them directly into the plant root, along with moisture as well. So they're a really key organism. But how does this all work? Well, really it's mainly driven by food. And so we've got exudates coming from plant roots, and we've also got organic residues. And if you could imagine that some of those organic residues are made up of simple molecules, for example, things like sugars, maybe some amino acids and things like that, relatively small molecules. Those molecules are small enough to be able to be easily absorbed through the membranes of microorganisms, so they can absorb them and use them as food. But there are also larger molecules as well. For example, if we've got some woody material, something like some sort of stiff recalcitrant sort of material, and those are made up of larger, longer chain molecules, for example, things like cellulose or starch or lignin, that kind of thing. They're too big to pass through that membrane, and so they need some further processing to enable them to become a food source for microbes, and so drive that microbial community in those soil processes. So if we have a look at a bit of a diagram about what a typical plant residue might look like and in terms of its makeup, you can see that it's made up of things like, for example, proteins and amino acids and sugars. There are things like pectins and waxes, and a large amount of it, but more than possibly 3/4, are made up of those recalcitrant materials like cellulose and hemicelluloses and lignin. And if you look at the headings there, you can see it's labelled what types of microbes, and this is in general, because in microbiology, things aren't always black and white. There tends to be shades of grey, I guess, like in a lot of areas of life. But in general, so bacteria are the things that are able to most quickly utilise those more simpler compounds, and those more complex things like cellulose and lignin are the food for fungi. And largely that's because fungi possess the organisms that possess enzymes or produce enzymes that are able to break those things down. And again, some bacteria also possess those enzymes, but by and large it's fungi that are able to do that. And so if we compare that with how fast or how slowly those residues break down, we can see that those fast things are able to be easily broken down by bacteria, and fungi break down those more recalcitrant things slowly because they need to act on them with enzymes. So there's another tip for practical management. If you're looking to, for example, if you feel or you've had, for example, a measurement that says that, oh look, your soil fungi are too low or your bacteria are too low, here's a little practical tool, actually a very useful tool or a key to work out, well, how am I going to encourage those organisms that perhaps are a little bit out of balance, or maybe they're too high. So what am I going to cut back on to reduce those? So there's a bit of practical, you can apply that to practical knowledge. What do they like to eat? They're a bit like us. They like a feed, and so the food source that's available for them is a key driver as to what sorts of microbes are going to be there. And so what can this soil microbiology do? Well, people that are actively managing their soil microbiology, for example, their soil health, tend to find that their soil systems tend to come into more of a balance. So they tend to lose that kind of good versus bad, boom versus bust cycle, all other things being equal. Rainfall, of course, is important, but all other things being equal, they tend to get a more balanced and even production cycles, which is something that you can take down to the bank then. Maybe it might not be, you might not get the highs that you do in the boom times, but you don't get the lows that you get in the bust times. And so that's something that you're able to take down to the bank, and your bank manager is probably going to really appreciate that, having a more even cash flow. So they're able to improve soil structure, for example, by increasing the percentage of water-stable aggregates in the soil. Particularly mycorrhizal fungi are a key organism in that. Because of their hyphae, they're able to glue things to bring things together in a net, and they also produce a substance called glomalin, which is a sticky kind of compound that acts a little bit like a soil glue that holds those aggregates together. So mycorrhizal fungi are also a really key organism in soil health and in soil structure. And in fact, if people were to ask me, as they sometimes want to do, what would be the single most important microbe group in the soil? If I was going to measure a microbe group, what single group, I could only measure one, what would it be? And I would say mycorrhizal fungi, because they are key to so many things, improving the soil structure, which remember is the kind of foundation for all of these things. So improving the soil structure, then you can make a home for other microbes, and the system can build and function and go on. So they're a really important microbe to have. And so what about some of the types of these microbes? So we've got, for example, we talked earlier about some of these different types. The main types, we've got bacteria, of course. We've got the archaea, relatively newly discovered and not so much known about them because they tend to be unculturable. The typical way of, a classical way of studying microbes is we grow them on agar plates and see what they do and treat them with various substances and compounds and find out what they do. These things are archaea, mostly unculturable, so not as much known about there, but they do increasingly thought to play some important roles in the soil as well. And we've got our fungi, of course, as well, and then other microbes as well, also doing various things. And so if we think about the soil as being a bit like, this could be Pacific Bay Resort here, and a little bit like us, we're all in our different rooms, and we all probably like different things. And so there are different types of microbes, and they prefer different types of environments. And so just to make it hopefully a little bit interesting for people to understand, we've got a type of bacteria that we like to call the party animals. Actually, in scientific terms, they call them the R strategists. But that's really pretty boring, so we're going to call them the party animals, and they like sort of moisture-rich, nutrient-rich situations. They like sugary things. So in our soil hotel, they're down by the pool, sitting in a nice shady spot, in a warm shady spot by the water, sipping on some sort of sugary drink with a little umbrella in it, just having a great time. Then the other type of broad type of bacteria are what are called the K strategists. But again, who's going to remember that? So we like to call them the quiet achievers. And they're the type of people that you might find in a resort. Their idea of a holiday is being up kicking back in the room and reading a book. And that's their idea of having a perfect holiday. That's the idea of a good time. And then up here on the roof, where my pointer is, oops, gone too far. Where's the party? There it is. There's the party. Up there on the roof, we've got fungi, and they're just doing something completely different altogether. It's up on the roof. They're completely different. Don't know what they're doing. Completely different to these guys here. And so these guys tend to, for example, these R strategist bacteria, they like these conditions, but you know, when the booze runs out and the pool dries up and the sun goes down, those guys disappear. And so they're relatively what you might call, they're just around for the good times. And so when times get tough, for example in the soil, it gets a bit dry, a bit hot, conditions are a bit adverse, they disappear. But these guys are able to withstand they can hang on longer. They're able to withstand those conditions. They just persist and hang on. That's why we call them the quiet achievers. And that's mainly due to differences in their cell walls. These guys are a lot tougher cell walls and some other strategies that keep them hanging on. For example, in dry conditions, they can still perform functions in the soil, whereas these guys are packed up, gone home, sleeping off a hangover, whatever. And so a bit of a key here is diversity. So the more diverse range of microbes that you've got in the soil, the more likely it is that there's going to be at least some of them that are able to carry on those four main soil processes that we talked about. If the diversity of your microbes is limited, that is also limited when, for example, you've mainly got these R strategies, these party animals here. When dry conditions come along, these guys disappear. There's nobody to do the work. That's kind of a gross sort of generalisation, but that's the point. And so diversity is another key point as well when managing soil microbes. So that's just an example. This is one of the party animals. This guy's Pseudomonas fluorescens, which is well known as a phosphorus solubilizing microbe. And these things coming out of his backside there, you might see these kind of cord-like things there. That's what are called flagella. And they're literally like little whip-like structures. And these things are kind of literally like outboard motors for bacteria. And so, and we've been able to see those using scanning electron microscopes and looking at these things. They're literally molecular motors. They're literally motors. They've got all the components of a motor. They've got two gears, forward, reverse, proton motive force, water cooled. They 10,000 RPM. They are literally like outboard motors. And so to me, it's fascinating that all this stuff is going on underneath our very feet, and we don't even realise it as we're walking along. Then we've got actinomycetes. So this is an example of one of the quiet achievers, and these guys look a little bit, to me, I think they look a little bit like instant noodles when they're all together. And an interesting thing about these guys is they're actually bacteria. They're called... Now the proper term is Actinobacteria, but the name actinomycetes persist. And when they were first discovered under the microscope, they were called actinomycetes, which generally refers to fungi, because they live in colonies in these filaments. And so they're not like your typical bacteria. They actually live all together in these filaments. And so when they were first observed, they thought, here's a new type of fungi with its filaments. But they're actually bacteria. And they're also responsible for that smell when it's rain. Who's been dry for a while, you get a shower of rain, you get that kind of rain smell comes up. Some of it, at least, is going to be a volatile compound produced by these guys called geosmin. Again, who comes up with these names? But geosmin is what it's called, and raindrops hit the ground. They've built that up over, it's been dry. So remember, these guys are the quiet achievers. They can keep working in drier conditions. It's been dry. They've been producing this compound. Rain falls down, volatilizes into the air, and we can smell it. And then we've got our fungi. And just like bacteria, there are different types of fungi. So, for example, here's one that consumers of fizzy beverages, popular fizzy barley-based or grape-based beverages would be familiar with, yeasts. That's what they look like by eye and up close. And they're relatively immobile. That is, they can't move. So they relatively don't have a lot of control over what they're doing. And another main group of fungi are the moulds. And so these moulds have a little bit of an advantage over yeasts in the fact that they can actually, if they're missing out on some food, they don't have any food, they've run out of food where they are, they can actually expand to find some new food sources. But again, their growth is limited in dry conditions. And then the next lot could be a group we could call a filamentous fungi. And so these are microbes, these are fungi that form filaments like those white. Everybody will have turned over some leaf mulch and seen those white filaments. That's these guys. And they can rapidly explore an area to find food sources. So they can actually grow out and find their food sources. And they can remain active in dry conditions because they can grow out and find some food. They're not stuck in the one place. They can even go out and find some moisture. And one of these types of filamentous fungi are Vascular mycorrhizal fungi. And these are kind of a unique type of fungi. And they're a mycorrhizal fungi, but they're a type that grows in probably about 80% of land plants and probably just about all food crops except for things like brassicas and lupins and some other exceptions as well. But these guys live in there, and this is actually one of the first micrographs that we took when we started our business about 10 years ago. This is actually from a real sample from a wheat crop. And in this picture you can see these kind of oblong structures here are the cell walls inside the root of the wheat plant. And these blue sort of knobbly lines are the filaments, or the hyphae they're called, of the fungi, growing in through the root cells of the plant. And then these fuzzy-looking things inside the cells here are what are called Bascules. And that's what the A in VAM is, if you've ever heard the term VAM, it stands for Bascules. So these things are the Bascules. And they have an incredible surface area. And the word Bascule comes from, I think it's another Greek word is a combination of two Greek words. One, arbor, meaning tree, and scule, meaning tiny. So they're literally tiny trees. That's what they look like. When people first saw them under the microscope, they look like tiny trees. And they've got an enormous surface area. And this is where the nutrient exchange between the mycorrhizal fungi and the plant takes place. So mycorrhizal fungi takes up some nutrients, gives it to the plant, and simultaneously the plant is giving the mycorrhizal fungi some sugars, and the mycorrhizal fungi is absorbing them. And that's the interface, the place where that all happens. And this blob here is what's called a vesicle, and that's where the V in VAM comes from for people who call them VAM. This thing's a vesicle, and it's like a storage sack full of lipids. So when the fungi's getting its carbohydrates and its sugars from the plant, sometimes it's got more than it needs, and it stores them up in these vesicles, converts them into lipids like fats, and stores them in these little storage sacks called vesicles. And it can use them later when it's time to make spores. All of a sudden it needs a lot of energy, so then it'll convert these storage sacs into spores, and so it can use the energy to make spores later on. But not all of these mycorrhizas form these vesicles, which is why technically now they're just called arbuscular mycorrhizal fungi, because only a percentage of them, of species, form these vesicles. Most of them don't. All right, so these mycorrhizal fungi, I suppose we've talked a bit about them, but they're unique in that they must live in association with plants. So these arbuscular mycorrhizas, or VAM, they have to have a living plant host. They can't survive without them, although of course they make spores, which when a plant comes along again can germinate and infect the plant, and so on they go. And of course we mentioned before that they produce this glomalin, which can glue soil together. Just how are we going for timekeeper? Another 10 minutes. All right. Okay, so we'll zip along. Yeah.
While you're taking time.
Yeah.
Those little vessels, they're lipids, you said. Do you know the relationship in the soil with algae and omega-3 in those lipids, or coming into the plant, or where that sort of...
The lipids are actually made by the mycorrhizal fungi, so they take up carbohydrates, and when they've got more than what they need at a given time, they'll, bit like our livers, convert it into a fat, or in this case a lipid, and then they'll put it in that vesicle and store it there for later on. Yeah.
Will that be transferred higher up the plant for animals to access?
No. What happens is when the fungi, usually when they go to make spores, so the question was what happens to those vesicles? Do they get taken up into the plant? When it's time for the mycorrhiza to make spores, usually all of a sudden it's time to reproduce, a bit like us. Oh, I need a lot of energy. And so what it'll do is draw on those lipids then and use that energy to make the spores. So that's as it's understood at the moment anyway, but good question. All right, so moving along a little bit, if I can get my notes sorted here. Yeah, sure.
Is that last one the only, the band guy, is he the only one that produces the globulin out of the mycorrhiza?
Yes, yeah. The arbuscular mycorrhizas are the one that produce the glomalin. Yeah. So there are other types of mycorrhizas that live in association with trees called ectomycorrhizas, and they can live by themselves. They do produce some glomalin, but by and large, it's these guys that do it, and probably because of the way the hyphae grow in the soil, and they tend to get stimulated by soil particles and so on. Without going into, anyway, I won't get into the details, but see me later if you want to talk more about it. I can talk about it for a long time, probably longer than you'd like. And so that brings me to getting to the main point here. So we've got our big four soil processes, which are our residue digestion, nutrient cycling, soil structure maintenance, and pest and disease regulation, from which all of our soil health properties come from, all those things but today just focusing in on one of them, residue digestion, and this was it's kind of linked with the topic of the day, and so this residue digestion is really the soil carbon cycle. And so really this is where the breath in the soil comes from. My brief was living, breathing soils. And so this is the soil carbon cycle is really living, breathing soils. And just due to our interruptions and time constraints, I'll have to zip through this a little bit more quickly than I would like. But basically we start off with photosynthesis. That's the ultimate start of the soil carbon cycle. We've got photosynthesis. And then out of photosynthesis, we've got respiration, which is the opposite of photosynthesis. So in photosynthesis, we're taking energy from the sun, we're taking light energy or electromagnetic energy in the form of light waves from the sun and photons, and we're turning them into chemical energy. We're capturing that energy and storing it as chemical energy in the form of carbohydrates. And then the opposite, that's a good storage system. And then we can shunt that, or the plants can move that around and store it for later, etc., etc. So then we've got respiration, which is the unlocking of those carbohydrates. And so this is where the breathing part comes in, because plants, just like us, breathe, and microbes breathe. And so it's estimated that about half of the annual photosynthetic carbohydrates are respired. So probably 50% per year are still stored in things like lignin and carbohydrates and other residues, and about 50% are respired. So half of that carbon that's photosynthesized is breathed out in the soil. And so here's a busy little diagram, which in future I hope to improve upon. But basically it's outlining the soil carbon cycle. And because of our limited time, I'll just kind of skip forward to a simpler version. Skip forward a few slides to a simpler version of that. And so one of the main points I wanted to make was about this carbon cycle is that there's really two fates, or two places that this carbon dioxide captured through photosynthesis can end up. There's two places that it can end up. And one is after being processed by microbes, degraded, it can end up either going back as carbon dioxide through the process of respiration, that's a natural process, to be taken up again by plants and cycled. Of course, this is why we call it the carbon cycle. But the other place that it can end up is in humus. And we've already heard some good and very interesting comments about humus earlier on this morning, which I was very happy to hear. And as was being said this morning, the humus molecule is very complex and has an enormous surface area and lots of bonding points. And we've already covered today pretty well the benefits of humus in terms of water holding capacity, nutrient storing capacity. So this is a really desirable thing, and I probably don't have to convince many people here. But probably one of another take-home message for today was about how we make humus. So we know we want humus. How are we going to make it? And of course there are many practical ways, that we could go about that. But what I want to do is just kind of lift off the lid of the black box of how humus is made, if you like. Just peel back that layer so we can look in and see what's actually needed to make humus. And then if we can sort of start to get an idea of how we can manage some of those things that we need to make humus, well, we can make more of it if we get it right. So basically, there's a lot of interactions, and it's basically breaking down those residues and reducing the amount of oxygen in those carbohydrates. So carbohydrates have got oxygen in them, basically getting rid of that and concentrating that carbon with other things. But it also requires the right amounts of carbon, nitrogen, phosphorus and sulfur, and also some clay and some silt and micronutrients to kind of build that nucleus around. And in order to stabilise a tonne of carbon in humus, and this is some Australian research that was conducted in the last decade or so mainly by a fellow called Clive Kirkby, and found that right around the world, humus has a pretty constant carbon to nitrogen to phosphorus to sulfur ratio. So if we can match, if our soils have the right amounts of carbon, nitrogen, phosphorus and sulfur, that means that it's a lot easier for that humus molecule and the microbes who are building it to actually find that stuff. They don't have to fight and get it from elsewhere. They can find that stuff and build that molecule. So they're some of the things we need. And of course, this is a biological reaction, so we need some respiration, but not too much respiration, because according to our diagram before, we've got carbon dioxide going off is one place the carbon can go, and humus is the other place. So the more carbon dioxide we've got going off the more carbon we've got being respired and going off as carbon dioxide, the less is available to turn into humus. But the catch is that it's actually a microbial process, so you do need to have some respiration, otherwise the process doesn't work, because those microbes need to breathe. So I suppose we've got about 2 minutes left. Right, okay. So I guess one of the key things here is that we're able to measure some of this stuff, and I'm not able to go into the detail that I'd like, but we're able to measure that, and so we've come up with these key indicators of measuring this, and so then calculating how likely your soil is in its current state, the potential of it to build humus. And so if you're looking to build more humus, you can find out, well, where am I at now, and what sort of things maybe I can address to make more of it, and then you're able to make management decisions based on that. There's a couple of people snapping some photos there, which is good. And so just because I went to all the trouble of it for setting up, we're going to just fast forward quite a few things here. There's a few case studies, and if you're interested in more of this information, I'm happy to provide it to you later and talk with you about it. But just to fast forward to now, I had a video, and just before we put that video up, which goes for a minute, so hopefully we'll have time to do that. So who is interested in being able to almost, let's say, instantaneously or in real time, start to measure some of these, for example, soil health parameters in their soil? Who would be interested in doing that? All right, quite a few people. Well, we've formed, over the last 2 years, we've had an agreement with a Canadian company that's making a handheld monitor that's able to measure using infrared the right spectra in the soil, and that we're able to correlate back to these microbial parameters and soil health parameters that I've just been talking about. And so using that, you would be able to get information pretty much in real time without having to send off things to the lab. And look, we love the lab work is great, and you probably never get something that's as precise as the lab in the field, but in terms of being able to have instantaneous measurement, one of the things with the lab is you have to. It's very complicated. It needs expensive equipment, and you need highly trained people that like to be paid a lot of money in order to work with it. So we're thinking this is often a barrier to people making more use of this information. So we've seen this as a way to make this information a lot more readily accessible. And also it can measure other food properties as well. And so we've just got a short video of it. This is going to be available in the first quarter of next year. We spent the last 2 years correlating it to these soil health properties that I've been talking about. And so the first quarter of next year it will be available. If you're interested, let me know. I'll put your name down, and if and when it's ready, I'll get in contact with you, and you can perhaps give it a try. Well, that's probably enough for me, and any questions, grab me later on. I'm only too happy to talk more later on. Thank you.